Joe, have you met Michelle?
No.
Michelle is the vice chair for Cortex Club. She
Awesome.
helped set this up.
Great. Ah, there's Michelle. Awesome. Hey, Michelle.
Hi.
Thanks.
Hello.
A sweet picture.
It's Terero's neurons.
Hmm.
So I checked, I shouldn't be load shed, so that's good.
Okay. We're still having quite a few people in the waiting room, so I'll just admit for a little while, and also just wait for Butler as well. Or before we start.
I'm not stopping to wipe up the mess here. So you must drain your watches. I'm quite happy to do the dishes, so I just haven't had time to get there.
Right.
I haven't had time. I'm happy to do the dishes. I haven't had time to get there.
You have to do some strategic meeting every now and again.
Yep.
Someone hasn't done the dishes.
I haven't either.
Well, I think you should try for a bit of that.
Yeah, I know. Yeah, I'm still hungry. I'll have some on bread.
Well, that's quite a tall order versus.
Okay, we still have a couple people coming. I think we'll wait another 5 minutes or so before Dr. Butler comes through.
Ready?
There is Dr. Butler. Okay, I'm happy to start if you are, Michelle.
Yeah, I think it's good.
Okay, sweet.
Cool.
So hello, everyone. Welcome to this first ever collaboration between UCT Cortex Club and UCT Internal Medicine Society. So it's a bit of a first. My name is Aaron, and I'm the chair of the UCT Cortex Club, and I'll be chairing this event as well. Mary Ghosh, who chairs the Internal Medicine Society, is currently stuck with clinical duties at the moment. She isn't able to join us now, but she'll hopefully join us a little bit later on. Deputizing for her is Michelle, who's actually the vice chair of both our societies. And I'd like to give a big thanks to her for helping put this event together and bring this collaboration to fruition. So the order of events for today will be as follows. Dr. Joe Raimondo will speak about epileptogenesis and how seizures arise in the brain. Thereafter, we'll do a short Q&A. Then we'll have Dr. Butler give us a clinical approach to seizures, again with a follow-up Q&A. Thank you very much to our speakers who will kindly share their knowledge and research with us. So just to set some ground rules, can all the guests please mute their microphones and switch off their cameras for the duration of the 2 talks? This is just to avoid distractions for the speakers and to also make the presentations as clear as possible. We would like you to please refrain from asking questions during the talk and rather just leave them until the end. If you have a question that you don't want to forget, just type it in the chat and the speakers will answer them after the talk. And just to let you know that this lecture is being recorded. So if you have questions and want to switch your camera on, just remember you are being recorded. Okay, so I think that kind of just about does it for the ground rules. So I'll start off by introducing Dr. Joe Raimondo. It gives me a very great pleasure to introduce him. Joe is a leading neuroscientist, both in Africa and the world. And much of his work has focused on this very topic of understanding the mechanisms of how and why seizures arise in the brain. And he began the first electrophysiology lab in Africa here at UCT and has developed the lab over time into a leading epilepsy unit worldwide, with postdocs, PhDs, masters and honors students all working under him. And I feel very, very lucky to be doing my thesis under him as well. So today he'll be speaking about epileptogenesis from a basic science perspective. So without further ado, I'd like to hand over to Joe.
Cool. Thanks, Aaron. Hope everyone can hear me. It's really awesome to give a talk for the Cortex Club. Yeah, Cortex Club is something kind of pretty close to my heart. I joined the Cortex Club in Oxford a little while ago, and it's so awesome to see it's flourishing here at UCT and in Cape Town. So I know Aaron said don't ask any questions for the 30 minutes, but if you do like have something you don't understand, just type it in the chat, and I'll keep an eye on the chat, because I mean, it's really awesome to get a little bit of kind of feedback as you go along, and if something's super confusing, it's nice to kind of get a sense of what you'd like to know more about. So yeah, I'm going to aim to talk for about half an hour, and we'll go from there. So hopefully by the end of the talk, you will have learned a little bit more about epilepsy, understood it a bit better perhaps, and also hopefully understood a little bit more about how the brain works as well. Obviously, those are two like super huge topics, but we're going to meander around both of them and hopefully learn something together. Okay, so this is the talk outline. I'm just hiding some stuff on my screen. Right, so this is the talk outline. We're going to briefly kind of start with why you might be interested in studying epilepsy. And I'm going to give you a little intro on neurons and networks. Not all of you come from a kind of strong neuroscience background. And then we're going to talk about what epilepsy is, what actually happens during a seizure, what's happening to the neurons and the networks, why our brains are actually prone to seizures in the first place. And then I'm going to end with a little bit of a deliberation about why seizures start and epileptogenesis. Cool. So the reason why I study epilepsy is it's a really awesome disease to kind of use as a lens to understand how the brain works. You can learn, hopefully you'll see during this talk, you can learn a lot about how the brain works. And at the same time, the more we understand about how neurons and networks behave and how those work, the better we actually understand epilepsy and can improve treatment one day. And as a kind of good example of someone who was studying epilepsy and trying to treat epilepsy, how they contributed to neuroscience and how the brain works, a really great example is a Canadian neurosurgeon called Wilder Penfield. So he was a neurosurgeon and neuroscientist, and he kind of pioneered epilepsy surgery to treat epilepsy, where you would try and pick out the part of the brain that's causing seizures. This is him, and you can see he's kind of labeling little bits of the brain here with different little labels. What he did was take a little stimulating electrode and stimulate little parts of the brain. And when he did that, he had his patients conscious as well, so they could tell him what they were experiencing, what they were feeling. And when he did that, he made a huge contribution to neuroscience in terms of kind of describing that actually function is localized in cortex. There's been a long kind of discussion or debate about whether you can focus specific functions to different parts of the brain. And by stimulating different parts of the brain and seeing what happens, as he did in his epilepsy surgery, he discovered these sensory and motor maps. Here, this is primary motor cortex, and he found that if, this is actually sensory cortex, sorry, he found that if you kind of stimulate the parts of the brain, you would perceive a sensation. So this would perceive a sensation of the index finger. And there's a kind of beautiful map of the whole body that's mapped out over sensory cortex over here. And you can see different parts of the body have bigger areas allocated to them. So if we rent really good sensation in our hands, we've got a lot of brain area allocated to that. And so an important thing also to know is that kind of brain activity passes through these maps, not across them. And an interesting phenomenon in epilepsy, when seizures, is something called the Jacksonian march, which is where you have a focal seizure starting somewhere, and the seizure activity just spreads across the cortical surface. Kind of activity is spreading in the wrong way. It's just spreading through adjacent areas of cortex. And this happens in motor cortex. A patient kind of can start with twitching of the thumb, then the index finger, then the middle finger. And as you can see, the different muscles would begin moving as the seizure spreads. And I think there's a neurologist called Hewlings Jackson who noticed this in his wife, amongst other people. So this is another cool example of seizure spreading through the brain, confirming or showing us that you have these motor maps laid out across cortex. Cool. So there's a little interesting tidbits about why we should be interested in epilepsy. And I'm going to give you a super quick intro on neurons and networks. So the kind of dogma we use in neuroscience is that this brain cell, the neuron, is really the most important and basic computational unit of the brain. And if you take a neuron and you stick an electrode on it, which is what we do in the lab, here's a little picture of what that looks like. It's a glass electrode lowered onto the surface of a neuron. And when you do that, you can see these neurons experience this very rapid change in voltage across their membrane, which we call an action potential. And this action potential sort of represents the output of the neuron. It means it's sending a signal. Yeah, so that's a kind of fundamental property of the neuron. Now, whether a neuron generates one of these action potentials or not depends on the input that it gets from other neurons. And neurons can get both what we call excitatory and inhibitory input. So an excitatory input will make the membrane voltage more positive, and an inhibitory input will make it more negative. And kind of as the name suggests, if you have lots of excitatory input, they will help or promote the firing of an action potential in that neuron. Conversely, if you combine inhibitory inputs with excitatory inputs, the role of inhibition or inhibitory inputs is to prevent an action potential from being generated. Cool. So I know a little bit about neurons and the kind of inputs they get. And obviously, as we all know, neurons are connected to each other in networks. So at the top here, I've shown a little convolutional neural network and artificial neural network. And these are super popular these days. Google has lots of them, which your phone is connecting to, enabling you to do all sorts of cool things like translate sentences or categorize photos. Here's a drawing of a real neural network from Raimondo, an early neuroanatomist. And you can see these neurons all connected to each other. And neurons connected to each other enable these networks to do some cool things. And one idea I'm going to just kind of seed now is the idea of an engram, something that can represent a memory. And when I say engram, what I'm referring to is kind of a subset of neurons and their connections in a network representing something like a memory. For instance, here we have a lot of neurons, and the red ones we can think of as the active ones. And we can say if these 5 neurons are active, that's representing a cat, for instance. And so you could have the same network, and if a different set of neurons are active, that might represent something else, like Nelson Mandela, for instance. So that you can see how this network could encode different things by having different neurons active at the same time. Cool. All right, so that's the end of my lightning fast intro to the brain. So now we're going to talk about what epilepsy actually is. Butler will talk a lot more about this. So just to say that epilepsy is this disorder where you're predisposed to experiencing or generating epileptic seizures. And practically this is normally defined as having 2 unprovoked seizures more than 24 hours apart. And it's a very common neurological disorder. There's 2 famous people that you may know. This is Prince and John T. Rhodes. Some very prominent and very successful people have epilepsy. It's got a particularly high prevalence in Africa, mostly due to high rates of brain trauma and CNS infections. Okay, so that's epilepsy, but what is a seizure? Okay, so a seizure is, when we think of a seizure, it's really what you observe, or it's this sort of the clinical manifestation of some abnormal activity in the brain, and this is really excessive excitation and synchronization of a group of neurons. And I'll get a bit more specific about that in a second. So some neurons are being very active in your brain, and then the seizure is the clinical manifestation of that. So it could be the sort of uncontrollable jerking of your arms and legs, or perhaps a person may lose consciousness, or they might get some kind of cognitive or emotional symptoms. They suddenly might feel very anxious or scared, or have a sense of deja vu. The kind of clinical manifestation is often very tightly linked to the area of the brain that's actually seizing. Okay, so here's a little example of a mouse that's been injected with a proconvulsant, and you can see this is a electrical trace representing the cumulative activity of quite a few neurons. And these are like individual little seizure-like events. And you can see each time there's a seizure, the mouse is kind of jerking. And I think it'll have quite a big seizure here. You can see its limbs are twitching, and it's unable to hold its posture. So this is a kind of what a seizure would look like in a mouse or animal, and very similar in a human. Okay, so useful concept is just that seizures can be typically focal or generalized, and all that means is for a focal seizure, we think of the seizure occurring in a particular area of the brain. These traces represent electrical recordings from specific areas. And so this seizure is starting in a specific place. And if it's generalized, the idea is that it's affecting pretty much all of the brain. It's spread to include all of the brain. Okay, so I loosely alluded to the fact that the neurons are going a bit crazy. They were firing excessively. So during physiological activity, your neurons' firing rate is, depending on the neuron type, can be quite low. And the important thing is the neurons aren't all firing at the same time. You can see it's kind of relative to each other. They're firing kind of asynchronously. But during a seizure, for some reason, all the neurons start firing very synchronously, and all of them are firing. And that's kind of a bit of an important point. That's kind of still debated, and I'll talk a little bit more about that now. And this is the question of whether all of the neurons are involved in a seizure. When I mean all of the neurons, I'm referring to like if there's a seizure focus, are all of the neurons in that seizure focus involved in the seizure or not? And here's a little video we took in the lab to kind of give you a sense, a flavor of this. This is a slice of a mouse's brain. We've done some special genetic tricks to the mouse's brain so that all of the neurons are expressing special genetically engineered protein which fluoresces when calcium enters the cell, or fluoresces when the cell is active. So the brighter the cell, the more active it is. And you can see, and this is a looping video before, and you can see there's like some of the neurons are firing, they're firing at different times. And then at some point, pretty much every single neuron in this brain slice is active. And so what you're seeing here is the start of a seizure-like event in this brain slice. So it's kind of a cool image of what is happening in a part of the brain during the seizure. Okay, and just to kind of bring this home a little bit, I'm going to reference a really cool paper that was in Nature a few years ago. This paper wasn't about epilepsy at all. It was actually about, our meeting has been upgraded. This is great. This paper wasn't about epilepsy. It was about engrams or memories, as I referred to earlier. And they were doing some really fancy tricks where they could use special tricks and light to artificially recall or activate memories in a mouse. What they did was they had these genetic tricks where they could express genes in neurons that were active. Okay, I won't go into the details of this, but essentially, if they take away this drug called doxycycline, then during that period where the drug is not there, if a neuron is active, it starts to make a fluorescent protein, amongst other things. So, for instance, here is this slice of this mouse's hippocampus where they took away the doxycycline and they put the mouse in a cage and they gave it some sort of stimulus and it generated a memory, and they've managed to label all the neurons that were active during that time. There, you can see them here in green. So these neurons represent the engram for that memory, which is very cool. And they show later in the paper that they can activate that memory in the mouse kind of artificially. But one control experiment they did was they triggered a seizure by injecting the mouse with a proconvulsant. And then they looked at how many neurons were active. And you can see basically all the neurons here were activated during that period. And here's a little population graph. You can see during a memory, you might use like 3% of the neurons. But during a seizure, almost 100% of the neurons were active. So this is another kind of example that the seizure involves all of the neurons being active at the same time. So what is wrong? Like, what's wrong with having all the neurons active at the same time? Why might that be bad? So early on in the talk, I was talking about these engrams and how we can think of memories, thoughts being encoded by subsets of neurons being active. So say you have a little population of neurons, a proportion of them be active could encode something, and if different neurons were active, they could encode something else. So you could encode a whole lot of different things with the same population of neurons by just having different ones active at different times. And here's a little graph about the number of combinations, how many memories you could store in your population, depending on how many neurons are active at once. So as a kind of extreme of this, during a seizure, if all of the neurons are active, there's only, there are no combinations you can store. They're all active, and you can only there's just one thing that you can store, like all active. And similarly, if they're all not active because they're in some sort of post-seizure depression, you can also like store no information. It's only when you can have small numbers of them active so that you can recruit different neurons at different times to represent information. So I guess the obvious impact of that is that you can't do any computation, you can't represent different memories if the entire network is active. Cool. Okay, so that was a little bit about what's going on during a seizure. I'm going to talk briefly, I'm just checking the time, about why networks, and I'm going to refer mainly to cortical networks here, why they're prone to seize. Okay, so there are quite a few reasons why networks are prone to seize or have seizures in the first place, and I'm just going to refer to like 3 different things as kind of cool examples. I'm going to talk about Hebbian plasticity, recurrent networks, and some synchronizing influences. Okay, so first I'm going to talk about Hebbian plasticity, which is super important for learning and memory. Okay, so if a brain is able to learn stuff, which is obviously very important for surviving in your environment, the current idea is that there's some form of Hebbian plasticity happening. And referring again to this idea of engrams, I'm going to explain to you how Hebbian plasticity works. Okay, so imagine we have a little population of neurons, okay, and they're activated by an external stimulus that's activating these neurons. So here our stimulus is a little circle. And this causes those neurons to be active for a while. And Hebbian plasticity refers to a process whereby if neurons are active at the same time, they strengthen their connections with each other. Kind of phrase that's used is cells that fire together, wire together. So here these cells have all been active together, so the connections between them are strengthened. Okay. So this is the learning process. You're kind of strengthening the connections between these neurons. So that later you might not need that whole stimulus. You can have just a little part of the stimulus. Because the connections between these neurons are now so much stronger, just activating a subset of them is sufficient to activate all of them and reactivate this engram. So you maybe see a little part of the cat and you say, oh, that's a cat. You don't need to see the whole cat, for instance. So this is one of the reasons, an example of Hebbian plasticity being important for memory formation. You can see there's a kind of sort of immediate issue here, which is that if you have active cells, they're going to form stronger connections with each other. So now they're even more active cells, and they're going to form stronger connections with other cells that are just happen to be active at the same time. So you recruit even more active cells, and they all get stronger connections. And if you ever try and make a kind of simulation where you only have this strengthening of connections where neurons are active, rapidly all of the neurons start firing at the same time, and you have a seizure. I guess the take-home message is that just the fact that our brains are able to learn stuff and strengthen connections means that we're probably going to be susceptible to seizures. Okay, I'm also going to talk about recurrent networks. So recurrent networks are really important in the brain. And a recurrent network is where you have neurons that are connected to each other. They're recurrently connected. So this neuron is connected to that one, and this one's connected to, they're connected to each other. So what it means is that if some of them are active, they activate their partners, and those recurrently connect back to their partners. And so you can see that very rapidly, the more activity you have, the more feedback connections you have, that promotes even more activity, and you can kind of get this runaway positive feedback, which would cause a seizure. The reason why these networks are all over the brain is because actually recurrent networks are very good for pattern completion, which is one of the things our brains do very well. It's kind of a useful function. But you can see by themselves, these recurrent networks would be quite unstable. And then the last kind of thing that kind of makes us a little bit prone to seizures, there are many other things, but these are kind of 3 examples, are synchronizing influences. So for instance, if this network had to get a whole lot of input where all the neurons are activated at the same time, that might make them synchronize and all be active at the same time and connect to each other, and I guess easily cause a seizure. If sensory input happens at the same time, that's quite a synchronizing influence, so like a strobe light, for instance. The other kind of synchronizing influence that you can think about is this: if you are going to be, if we're conscious of certain things happening around us, or percepts, we need to bind a whole lot of sensory input together, thoughts together from different brain areas. So there has to be kind of rhythms that are generated where things happen synchronously, need to be bound together. And so there are processes which synchronize activity, and having synchronous activity, too much synchronous activity is really what a seizure is. Okay, so why aren't our brains seizing all the time? Well, because there are lots of sort of anti-seizure processes that are balancing some of these things I've mentioned. So for instance, I talked about Hebbian plasticity, but there are other processes that happen, things like metaplasticity and synaptic scaling, which ensure that connections between neurons don't just get stronger and stronger and stronger. In fact, what happens is if some connections get stronger, others have to get weaker to compensate. So there are all sorts of processes that keep that in check. And then another really important thing we have in the brain is synaptic inhibition, which I'm going to talk about very briefly here. So I mentioned it earlier. Inhibition is synaptic inhibition makes the membrane potential more negative and less likely that neurons generate outputs, fire action potentials. So if we had our recurrent network, it might be really useful to have some recurrent inhibition in here. So here we've got another neuron, an interneuron that inhibits its targets. Now you can see the cool thing about this is like the more active these excitatory cells are, they're recurrently connected to each other, but they're also recurrently connected to this inhibitory neuron. So the more they excite themselves, the more they excite the inhibitory neuron, which inhibits all of the neurons. So the more excitation you have, the more inhibition you have, and you can see things will stay balanced and keeps the network stable, which is cool. It's only a little bit of research I'm actually going to show you that I've been involved in, and that's that during a seizure, this inhibition can actually become excitatory, which is really crazy. So here we have a seizure. You see this neuron is firing action potentials very, very quickly. And what we're doing here is activate synaptic inhibition by puffing on GABA, which is the major inhibitory neurotransmitter. And what you can see is we do this every 10 seconds. And before the seizure starts, this inhibitory neurotransmitter is being inhibitory. It's causing negative shifts in the membrane voltage. What's kind of crazy is that once the seizure has got going here in T2 and T3, when we puff this GABA inhibitory neurotransmitter, instead of causing a negative shift, it's actually causing a positive shift that's triggering action potentials. It's become excitatory, which is and kind of exacerbating the seizure. And that goes back to normal after a while. So just to kind of show you what it might look like, if we had our inhibition stabilizing the network during a seizure, this actually can be subverted and also be excitatory and help keep the seizure going. So we've had a failure of inhibition. Cool. So you can see anything that might reduce inhibition, say you have less inhibitory interneurons or their connections are less strong. All of those things would make the network more unstable and more likely to have a seizure. Yeah, so in the last 5 minutes, I'm going to just do some musings on epileptogenesis and why seizures kind of start in the first place. And it's going to be kind of pretty high level and abstract, but hopefully it's kind of useful to think about. Okay, so I'm going to try and describe the activity in a network of neurons, or say an area of the brain, with these little scribbles. My drawing skills aren't very good. So what we're doing here is you could plot, make a multi-dimensional graph with on each different axis the activity of different neurons. Yeah. And what we're going to do is reduce that all down to just 2 dimensions, 2 kind of random dimensions, and we're going to plot the state of the network as this little purple ball as being somewhere on these arbitrary dimensions, which represents the particular activity state of that network in any particular moment in time. Also, of course, there are some kind of, there's a limit on the different kind of network states you can have, like which neurons are active and how active they are. And the kind of landscape of possible network states, I'm going to just draw here is this like squiggly line. And because of the way the networks are structured, most of the time the state of the network is in this kind of little basin or attractor, and we can refer to this as physiological activity. So different kinds of physiological activity that it always kind of stays in this area. There are forces that make it more attractive to be in this area, kind of like a little marble in a basin. But we know it's also possible to go into a different state, a seizure state, where the network will stay in that seizure state for a while. You can kind of think of it as these two basins or attractors. Okay, so for a normal person or a healthy person, their average day, including perhaps seeing strobe lights or something that kind of prompts their network activity to go into kind of weird activity states. Just do that again. Stopping my animation. You can see that the balls just hang out in that basin, but it doesn't ever pop out. Okay. But we know that even a healthy person can have a seizure. They just need the network activity to be pushed into such a different weird place that they pop over into this seizure activity basin here. So maybe they get electroconvulsive therapy, they have a super high temperature, which makes the neural activity go crazy, and they pop into the seizure activity state here, or you give them a lot of potassium, for instance. So it's certainly possible for a healthy person, given the right stimulus, to have a seizure, and they'll stay in that basin for a while, and then other processes will happen which bring them back to normal. So what happens during epileptogenesis is there's some sort of sequence of events that converts a normal-ish neural network into a network that's prone to seizures. So I've tried to kind of draw that here as a change in this landscape of possible network activity states. So you can see what's happened is it's now easier for network activity to pop over to this other stable attractor here on the right, and it's easier for the ball to stay in there for longer as well. So loads of different things that could cause this: changes in gene expression or trauma or infection. All of them are somehow changing the structure of the network. Maybe there are fewer inhibitory interneurons. Maybe plasticity processes are different. Maybe there are microglia that eat the inhibitory synapses rather than the excitatory ones. All sorts of things could shift this landscape of possible network activity states and make it more likely that you can have a seizure. And they can happen over different timescales. Maybe, I don't know, drink some coffee or take some amphetamines, and there's a neuromodulator that changes the network a bit, making it more likely that you can have a seizure. If we have an epileptic person might get the same stimulus, like a strobe light, that much more easily pops them into a seizure activity state. Right. So I guess the goal of anti-seizure medication would be to shift this network to make it less likely that it can go into seizure activity states. Ideal would be just to put it back to normal, but what most anticonvulsants do is obviously also shift the kinds of normal network activity states that you could have under baseline. So there might be cognitive side effects, etc., etc. So I hope this kind of just giving you a little flavor for maybe the the chaotic sort of random nature of how seizures start. You can't really predict exactly when a seizure would start, but you can kind of know when it's more likely or not if you understand whether the structure of the network has been perturbed. Right. So I think, yeah. Just to keep things short, I'm going to conclude here and hopefully remind you that epilepsy is a disease that kind of emerges from the necessary structure of brain networks, and that's understanding a bit about epilepsy teaches us about the brain and these networks, and the converse obviously applies as well. Understanding the brain will hopefully help us improve our treatment of epilepsy. And that is it for me. Are there any questions? No questions?
I have a quick question, Joe.
Yeah, sure.
If you had all the money in the world, where would you invest in terms of the potential finding out what's actually going on with seizures? Put your money in one place.
What would I do with all my? I think this is that Mbizo question we asked all our applicants. Yeah, what would I do? I think trying to think in terms of like a useful strategy to like actually cure epilepsy, or what would I do if I just was curious and wanted to understand how things worked? Like, I mean, if I had lots of money, I would buy very fancy microscopes that would let me kind of observe each neuron in a behaving animal whilst in the run up to and during a seizure, which would be really, really cool. And then you would obviously need lots of fancy computers to try and analyze what the data you get. I'm a kind of, the way I like to do science is quite like observational. You're meant to have hypotheses and then like prove those right or wrong. But I kind of get the most satisfaction from kind of just observing things and then trying to work out what's going on and work things out from there. So I'd probably use all that money to buy fancy toys to try to work out how seizures work. And then as we've done before, you can use what you find to inform better management of epilepsy. So we've done work on why seizures don't stop and thinking about the best treatment strategies you might want to use if you actually want a seizure to stop. We're getting some chat questions here. Excellent. Thanks, Emma-Louise. Ian's got a question. In the Nature Letters paper, where selective cells were activated by removing doxycycline, how did they manage to select a specific hippocampal engram, such as the specificity fear memory engram, if I understand correctly? Yeah, I'm going to just share my screen again. Okay, so I guess the question is really how were like just specific neurons activated, or the specific neurons in the engram. So what I didn't maybe describe well was that what they have is a system where when they remove this doxycycline, if a neuron is active, there's something called the CFOS promoter, and the CFOS promoter is kind of turned on only when a neuron is active. It's kind of like one of these genes that if a neuron's firing a lot, it gets expressed. And so they've put that promoter in front of this element that responds to doxycycline. So basically, if this is normally inhibited by doxycycline, but if doxycycline is taken away, when the neurons are activated, the CFOS promoter drives the expression of a whole lot of stuff, channel rhodopsin and a fluorescent protein. So this is only going to happen in the neurons that are active during the time that there's no doxycycline. So they stop feeding the mouse doxycycline, then they put it in a cage where it establishes a memory because they shock its feet and play a sound, and the mouse establishes a memory. That's kind of how it works. Hope that's clear. Yeah, cool. Yeah, feel free to unmic yourselves if you want to just ask a question directly. Japan has asked if there's any difference in how generalized seizures may differ by focal onset ones, and I have absolutely no idea. I guess there's a conceptual question. We know a seizure that starts at a specific focus definitely starts there and then can kind of spread. And it's often unclear why it actually starts there. There must be something about the neural network that makes it more likely that it'll start at that focus. But how does a generalized seizure start? Because those are seizures that appear to just start in the whole brain at once. And there's maybe a philosophical question is whether they actually start in one place and just propagate so fast that you don't actually see where it starts, or whether they truly start all over the place at once. I actually don't know the answer to that, unfortunately. Okay, that's a great answer. Christina's asked a question about pattern completion and how this might make our brains prone to seizures. So I mentioned that we have recurrent networks where neurons are connected to each other. And recurrent networks are particularly good at completing a pattern. So what that means is, say, it's an example, like in that, let me just share my screen again. So in that example of an engram, of what an engram was, 1 second. Yeah. So this is a recurrently connected network. You see all these little neurons are recurrently connected to each other. They're connecting to each other. And this is a great example of pattern completion. Here's a pattern, a circle that's activating all of them. And if you just present part of the pattern, because the neurons are recurrently connected, it's able to complete the pattern in a way. The representation is able to complete itself. So that's a property of recurrent networks. They're good at pattern completion. So maybe you hear part of a song, but because you've heard the song so many times before, the connection between the neurons that represent that song is quite strong, so that you just hear the intro to the song, and then you can kind of complete it in your head because you can reactivate the whole rest of that engram that represents that song. Not sure that makes, hopefully that makes a little bit of sense. Cool. Right. Stefan's got a question. Where do you think doctors in South Africa fail in terms of treating or diagnosing epilepsy? I think that's got to be a question for Dr. Butler, I'm afraid. I have absolutely no idea. I'm quite far from the clinical interface. Yeah. Cool. So I think that's all the questions. Thanks all for your really excellent questions, and hopefully we're still running on time. Haven't delayed you too much. Thanks, Aaron.
Thanks, Joe. That was great.
I'm gonna stop there.
Yeah, and thanks for all the questions, everyone. Those are some interesting questions there. I think that last one, Stefan, we'll save for Dr. Butler after his talk. So thanks very much, Joe. I think everyone there found it really informative.
Cool, pleasure.
I think I'll hand over to Michelle now just to introduce Dr. Butler.
Thanks, Erin. Thank you, Dr. Raimondo. It's my pleasure to introduce our next speaker. Dr. James Butler is a widely respected neurologist who currently works at Constantiaberg Mediclinic and is also an honorary consultant at the UCT Neurology Department. He completed his undergrad and postgrad at UCT, followed by an epilepsy fellowship at the University of Western Ontario. He currently only sees people who present with seizures, and so we could not be more pleased to have him present a clinical approach to seizures. Over to you, Dr. Butler. I think he is.
Very much. Can you see me? Can you hear me?
Yes.
Good, good. All right, thank you. Yash, I thought I will, or what I'll do is I'll share a screen with you, some PowerPoint slides, and just hopefully that appears. You see that?
Yes, thanks, Dr. Butler. We see it.
Okay, great. So, yeah, you know, the term epilepsy is one that's obviously widely used. It appears in pretty well every textbook, either on the front of textbooks or in chapters where the neurology books which deal with the epilepsy, or what's called epilepsy. But I personally think it's conceptually much more useful to think about it as the epilepsies, because as I'm going to show you, there's a great variety of different conditions. They manifest in very different ways. They have many different causes. Sometimes you don't even have symptoms at all or signs at all. And so in this rag bag of conditions, I think one best think about this as the epilepsies, which I think just emphasizes the spectrum and the diversity of what we're talking about. Now, Joe's told you a little bit about what an epileptic seizure is, and the definition is one that's still physiologically based. As he explained to you, there's this electrical storm, which is the terminology I use for patients. It's really important actually to explain to patients what's going on with them, and we can talk about that a bit later. But the main reason that it's still physiologically defined is that there are two, in very broad terms, there are two types of seizures that we see in clinical practice. And the one we call epileptic, and the other are non-epileptic. And there are all sorts of things that can mimic epileptic seizures, but none more so than what are called non-epileptic seizures, which represent part of the spectrum of conversion disorders, a group of conditions where, a very interesting spectrum of conditions which we don't have time to talk about, but which you don't have this electrical storm during the brain. And these patients present frequently to neurologists. They're frequently misdiagnosed as having epilepsy. They're frequently treated as epilepsy, sometimes for many years, sometimes for decades. And I've seen people even lifelong treated for epilepsy with medications when they don't have epileptic seizures. So the definition is physiological or neurophysiological rather than clinical. So when someone presents with a seizure, the first question they'll ask you is, why have I had it? Well, that's the question you might ask if you presented with one. And of course, the answer is there are lots of causes. It's largely a cortical process, although we've got lots of good evidence that the subcortical systems are involved in the genesis of both generalized and focal seizures. And in very broad terms, one can think of at a clinical level, and these are just three very big baskets to put causes into. There are those where there are genes involved, and more often than not, there are at least several genes involved in the genesis of someone with a genetic form of seizures. The old terminology was idiopathic. Then there are those where there's been some sort of previous insult to the brain, which might be acute, and then we call it acute symptomatic. And there's something, in some instances, there might be something that's happened to the brain in the past, in which case we use the term remote symptomatic. And then sometimes a person has a seizure and you've absolutely no idea why. You do a bunch of tests and they're all normal. Now, when someone presents for the first time with a seizure, this condition of acute symptomatic seizures needs to be considered, because as Joe has told you, you take any normal brain, mine or yours, and you subject it to sufficient metabolic stress, either deprive it of oxygen or glucose, or change its metabolic milieu in terms of the amount of sodium that it's bathed in, as it were, hyponatremia or hypernatremia, or diabetics in whom the glucose levels drop or where their osmolality goes up. People like that present with seizures, and we call those acute symptomatic seizures, which is distinct from a propensity to recurrent seizures, or in theory anyway, it's distinct from that. So we always want to get those out of the way. They're usually pretty obvious. Someone's acutely ill for whatever reason, diabetic or otherwise, or they've had a bleed in their brain, or they've got meningitis, etc. Now, of course, some of those insults to the brain may have permanent sequelae, which may produce an enduring propensity to seizures. So when we see someone who presents for the first time with seizures, we try and get acute symptomatic seizures out the way. And then you have to ask, is there something obvious, so-called structural, and this is what worries patients and doctors, mind you, is there some sort of structural abnormality in the brain that can be seen on a brain scan? And something like 1 in 10 people will have a clinically important lesion. And the most common ones are actually very benign. They tend to be developmental, so-called malformations of cortical development or vascular malformations, occasionally sequelae of previous infections, and only occasionally are there brain tumors, which turn out to be nasty. So when we see someone, what do? Well, we have to do a scan, as you've just heard, and then to define and to start this process of working out the cause, we also do an EEG, and this quite often gives a clue, as I'll discuss later on, with the history into the possible causes. So, for instance, the various forms of genetic forms of epilepsies, which I'll tell you about in a moment, which may have characteristic EEG patterns. For instance, the so-called genetic generalized epilepsies have characteristic EEG patterns, whereas a particular condition in childhood called what used to be called benign Rolandic epilepsy or benign focal epilepsy of childhood has a particular EEG pattern, and likewise one which starts in the occipital region. So the EEG is useful in terms of that. Now, for years, we had no decent brain imaging in the years of Wilder Penfield. CAT scans eventually came along, and they disclosed some causes, but not all. And this is a CAT scan that you see of someone who it was reported as normal. This is someone who presented to our hospital for the first time with seizures. And because it was normal, but his seizures suggested right temporal involvement, I did an MRI scan. And just to show you the difference in the technology, this looks like a bit like a picture of the Loch Ness Monster. And the great detail and the greater detail you can see on an MRI scan is immediately apparent. And this patient has a large brain tumor in his right temporal region involving the medial temporal region. Do we need to do blood tests for people who present with seizures? Usually not, unless they have suspected acute symptomatic seizures. And ditto, do you need to look at the fluid around the brain and do a lumbar puncture? The answer is no, unless there is a fever or someone has neck stiffness. Either of those might suggest there might be meningitis, or if someone is immunocompromised, in which case they can't mount an immune response and you might not get a fever or neck stiffness. So seizures are common things. I think something like 7.8 billion of us on the planet, about 1 in 10 are going to have a seizure in a lifetime. So these are common things, and they occur in all walks of life, in all professions. Some very famous people in South Africa are patients of mine, some very wealthy people. So this is not confined to any particular social strata or demographic group. It's extremely common. And interestingly, across countries, when incidence and prevalence studies are done, there are small differences which largely are thought to be methodological. That in itself is a topic in itself. Now, when you've had one seizure, what are the chances you're going to have another one? On average, a year after your first or so-called incident seizure, about 1 in 3 people who have a seizure will have another one in the next year. And if you don't have one in the first year, the incremental life probability is 12% by 2 years. So most people who have seizures or recurrence after their first seizure are likely to have a recurrence within the first 2 years, because beyond that, the incremental probability of having further seizures is very small. So the point being, most people who've had a seizure are going to have a recurrence in the first year, and by 2 years, largely everyone who is going to will have had one. Now, as I've said to you at the beginning, there are many causes, and the cause is important when it comes to recurrence risk. So if you've had a previous insult to the brain, then your probabilities are about 60%. Whereas if there's no obvious cause, these are the genetic and the ones without a cause, then about 1 in 3 people will have a seizure at 2 years. So cause is clearly important in prognosis, and that in itself is, as I've said to you before, there are many different types of epilepsies, and for each of them, in fact, there's a textbook written on the prognosis of epilepsies with a look at all these different types. This brings me to the next point about Joe telling you that the definition of epilepsy requires 2 unprovoked seizures at least 24 hours apart. And what I'm going to tell you is that this definition is utterly arbitrary, and it's arbitrary for a multitude of reasons. The first is from a prognostic point of view, and this is a very famous study published in 1998 in the New England Journal of Medicine, looking at the likelihood of recurrence after a seizure, a patient who's had a first seizure, and this is months of follow-up, and you'll see most of the recurrences happen in the first 24 months, and after that the curve flattens out at about 30 to 40%. This is in their particular study, it was 30-odd%. But notice, so it's not certain after 1 seizure that you'll have more. And the distinction when it comes to the definition of epilepsy, if you've had more than 1, the assumption is then that you're going to get more, but that's in fact incorrect. Because as Hauser and them showed very clearly, after 2 seizures, the likelihood of recurrence is only about 2/3 of patients. So the distinction between a person who's had 1 seizure and a person who's had 2 seizures, let's say a week apart or a month apart, in terms of the likelihood of recurrence, is not that different. I mean, 30% is a large amount, obviously, but it's far from certain that this person will have seizures who's had 1, and it's far from certain that the person who's had 2 also will have more seizures. So the point is there's an increasing probability, but not a certainty, and so the distinction is somewhat arbitrary. Second, as I've told you earlier, the cause is important. So if you take someone who's had 1 seizure, but who's got a grossly abnormal EEG and a grossly abnormal MRI, the likelihood of recurrence approaches the same probabilities as someone who's had a second seizure. And the third reason is that if you look at the genetics of the epilepsies, then you'll find that if you take a particular mutation, let's say a sodium channelopathy, and you'll find that in the family, people with exactly the same mutation, some people will have 1 seizure, others will have febrile seizures, others will have a combination, others will have a multitude of very bad forms of epilepsy, and some of them will have forms of epilepsy with developmental regression and end up severely impaired. So the person with a mutation, do they have epilepsy or not? You can see you can have all sorts of outcomes. So I prefer to think of it as someone who's had 1 seizure or more seizures, and think of the cause and think of the prognosis to reflect this diversity of causes, prognoses, and hence the need for treatment. Again, after 2 seizures, cause is important. You can see that if you've had previous insults to the brain, seizure likelihood is more probable. This is from Hauser et al. So it starts to become very likely after one remote symptomatic seizure that you're going to have more. And the question then becomes this: if you've had a seizure, should you treat it? So this has been looked at in several studies, and the bottom line is that this is a difficult decision for people to make, because, as I said to you earlier, if someone's had one seizure, only about 40% of people are going to have a recurrence at 2 years. So you're really tossing the coin when you're deciding about whether you take treatment or not. And then secondly, it's far from certain that the treatment will be effective, because generally you started on one medication at a modest dose. And so this randomized controlled trial showed that there was a reduction where in the control group where they didn't treat people, half of them had a recurrence. But those who went on to treatment, the likelihood of seizure recurrence was 25%. So the absolute reduction was 25% here, which means that you've got to treat for every 4 people who have a single seizure and you put them on treatment, only 1 person will benefit. 2 won't have seizures and 1 will have a seizure despite treatment. So we come back to the definition of this whole issue of recurrent unprovoked epileptic seizures as the definition for epilepsy, or the epilepsies. And in broad terms, at a clinical level, there have historically been 2 classification systems, those based on cause or syndromes, and those on seizure types. And seizure types gives you a clue as to what happens in the brain during these seizures. And this is sometimes confusing for people when you present medical students, etc., with the idea that there are 2 different classification systems. But really, in principle, the ones to understand what's going on in the brain, and the others to try and understand cause, or at least lump them together in syndromes, because that impacts on treatment. So you heard that, sorry, was that a question? So you heard earlier that there's this broad classification which has been present for decades, subclassifying seizures into those that are so-called focal and generalized. And this is useful at a clinical level in a very broad manner, but it's misleading if you really want to understand the physiology of seizures, because the term focal implies an onset in a very, very discrete part of the brain, which is virtually never the case. When you put in electrodes into across people's cortex and you study the areas of onset, there are regions of onset which are so-called focal, and that's what the focal epilepsies really are. And the areas of onset can be substantial, involving many square centimeters, 8 by 8, etc., of cortex at times. And then there are the generalized ones where, in principle, you have a seizure which appears on EEG and clinically, but on EEG in an abrupt manner throughout or through over the entire head, as it were. And so those are the broad classifications. Those that start in one region may become generalized, and curiously, those that start in a generalized manner, as inhibition kicks in, might become focal. So this, again, this is a broad, generally useful classification system, but like all classification systems, it has inherent weaknesses. So focal seizures are subclassified into those where patients are aware of what's happening and those where they're unaware. And in principle, when a seizure starts in part of the brain where there are certain parts of the brain, you can get symptoms which reflect the function of the underlying cortex. So Joe gave you the example of motor cortex, but of course, there are many different parts of the brain where seizures can start. And some of them, seizures can start and remain in those areas and remain clinically undetectable for long periods of time. So I was looking at someone, for instance, today, where we've got electrodes in his frontal lobe, and he has seizures that are 7 minutes long, electrical discharge there, and he continues interacting with everybody typing on his cell phone, etc., and functioning normally. So that's a relatively silent part of the brain. But when seizures involve parts of the brain where function is manifest, it produces symptoms, and the symptoms seizures most commonly start in the temporal lobe. So you'll get, say, symptoms that reflect temporal lobe function. They start in the occipital lobe, which is where vision is, you might get visual symptoms. If they start in parietal lobe's responsible for sensation, so you might get sensory symptoms. And frontal lobe seizures can produce movements because especially the posterior parts of the frontal lobes are responsible for movements. So you might get all manner of movements, including strange things like people running or jumping, etc., thrashing, kicking, bicycling, etc. Now, seizures that start in one area might then propagate and involve sufficient cortex to produce a lack of awareness, and those are then subclassified as focal unaware seizures. The seizures might originally start in a large enough area that you do not perceive symptomatology. And in some instances, seizures might start in one part of the brain, produce symptoms, and we know this because the patient pushes the trigger button and to tell us they're feeling something. But then when questioned subsequently, they completely, they cannot recall having had any particular symptom. And presumably what's happening there, or what we can infer from what our physiological studies, is that the seizures have involved the hippocampus or the hippocampal region. The memory for the aura is obliterated, and no memory is laid down. And sometimes patients are dragged into my office here where they have seizures that have just involved, let's say, the medial temporal region, and they are amnestic and they're blissfully unaware of the fact that they've had a seizure, and feel perfectly fine afterwards and argue with their spouse that there's anything wrong with them. So those are focal unaware seizures. It gets a little bit more complicated because the distinction between memory and responsiveness and awareness is something that is worthy of a discussion. But suffice to say, in many of these seizures, which most often happen in the temporal lobe, where patients are unaware, they may or often engage in various forms of automatic behavior, including mouthing movements, or fidgeting, or wandering, or staring, licking their lips. If it's in the non-dominant hemisphere, they might speak. And sometimes they are capable of quite complex behavior as well, of continuing to eat. I had one patient once who continued tiling while he had a seizure, etc. And more often than not, after these seizures, patients are a bit tired. So those are focal seizures. And then the generalized ones, as I said earlier, might start focally, or they might be generalized in inverted commas from the outset. So the focal ones in the temporal lobes, I'm not going to spend a lot of time talking about this, but the temporal lobes are responsible for various things. I mentioned memory, which sometimes you get disorders where there's amnesia, but sometimes there are disorders of memory where patients have particular memories that are laid down. Deja vu is something that many people experience, but when it's frequent, it's often a symptom of seizures, and that's a disorder of memory. And the idea is the seizure pathway is always the same, so you always get the same feeling or the same aura as it's described by patients. The limbic system is there, so the amygdala, fear is a common symptom, representation of the visceral system. So the most common symptom in temporal lobe seizures are strange epigastric feelings. And because memory is laid down in the temporal lobes, patients might have all sorts of disturbances of and recall either a smell or a taste or hearing something or seeing something. And just to make the point about how unique this is to the individual, and then I've got many examples like this, but when I was in Canada, at first was brought home to me by a patient who was in the garden on a Sunday morning, and he heard this little jingle from a radio tune that was called Gilmore's Requests, and it started with this little jingle. And he subsequently went inside the house and said to his wife, what was on Gilmore's Requests? It was a sort of phone-in program and people could play music. And his wife said to him, no, it's Sunday. It's Gilmore's Requests is on a Saturday. And he was a bit puzzled. And suffice to say, he subsequently had this little jingle and as his aura of his temporal lobe seizures repeatedly over many years. And the point about the reason I tell you the story is that I have no memory of what Gilmore's request, the jingle for Gilmore's requests is. So that aura was unique to him in the sense that he had it because that memory was embedded in his hippocampus and his medial temporal lobe system, whereas it's not in mine. And the idea is that the seizure runs the same old beaten pathway, and that's why you get the same symptomatology. I've told you about the other lobes, so I'm not going to spend any time on that. And then I don't have time. I've got lots of videos that I could show, but unfortunately there isn't time to just illustrate the differences between these. But the most common generalized seizure are, and one that most familiar to many people, are the so-called generalized tonic-clonic seizures, or what used to be called the grand mal seizures. And that's the old terminology where patients go stiff and shake. And those are generalized, and the EEG correlate is throughout over the entire head. Clearly, the motor symptomatology reflects involvement of motor cortex. Absent seizures, in contrast to the ones that I told you earlier about, the temporal lobe ones, where patients also stand and are unresponsive, typically for 30 to 90 seconds or that sort of time. Absent seizures are very brief episodes, what used to be called petit mal, where people just stare for a few seconds, unresponsive, and then continue normally, sometimes unaware that anything has happened, or sometimes just realizing they've lost their train of thought or have stopped in mid-sentence. And those, the EEG correlate of that is also generalized, so it appears over the entire part of the brain, but the dysfunction is largely prefrontal. And then myoclonic is a brief little movement involving typically the upper extremities, but it may just be the eyes or the face or the tongue or sometimes the buccal musculature, and occasionally it might be the legs. And when it involves the legs only, the person might just fall over. So one of the causes for people with unexplained drop attacks are just little brief myoclonic jerks in their legs. There the discharge is extremely brief. And then there are tonic seizures where patients just are briefly tonic or atonic, where they lose tone. And these are different descending motor pathways. As you know, some of those are excitatory to motor pathways in the muscles down the spinal cord, and the others are inhibitory. And so you either might have tonic activity or atonic. These more often than not happen in people with severe diffuse brain disorders, such as you might find in institutions. And they're the ones you will recognize in the street when if you see someone wearing a helmet, because they fall and they smash their brains and their heads horribly during these tonic or atonic seizures. Fortunately, relatively uncommon. Just to make the point that staring episodes might either be generalized seizures, so-called absences for a few seconds, or focal unaware seizures of, let's say, temporal or prefrontal origin, where people might have a warning. The staring tends to be longer, they're often automatic movements, and afterwards, there's more often than not, there's some fatigue or other symptomatology. So then we've spoken a little bit about what we could infer from the symptoms about what's going on in the brain. And then we just speak briefly about the so-called causes or syndromes. And here, as I said earlier, you think about genes, you think about previous insults to the brain, and previous insults includes developmental disorders of the brain. So during the early embryonic period, when cortex is laid down and the brain is formed, you can have various malformations that occur around that time. And while these might be completely innocuous in every other way, and they might be tiny and sometimes only visible under a microscope, they can cause devastating seizures and destroy people's lives. Nothing cool about that, I'm afraid. And then sometimes we don't know. As I just said, sometimes we have to infer from the investigations when all the tests are normal, there's no family history, you're down to microscopic abnormalities in the brain or genetic disorders. And this is what this rag bag presumably represents. Now, just to talk about syndromes for a moment, and I don't want to bore you with these, but it's important that we define these syndromes because more than anything else, it impacts on prognosis and it impacts on treatment. So what used to be called benign Rolandic epilepsy of childhood occurs in young kids, more often than not in sleep, and it's a condition which inevitably remits. Often kids have very few seizures, but even those that have lots of them, they remit within a year or so, but always by the age of 15. So they have an extremely good prognosis, and one doesn't want to end up over-treating children with this form of epilepsy. In the old days, many doctors elected not even to treat them. Then there's a condition called juvenile myoclonic epilepsy, which is a syndrome where you get 3 different seizure types, or 1 or 2 of them at least, generalized tonic-clonic seizures, absences, and obligatorily myoclonic seizures. And in contrast to benign Rolandic epilepsy, juvenile myoclonic epilepsy requires much longer treatment generally. The prognosis is a little bit more guarded. There's one very long-term study which suggests that about half of, from the Canadians, which suggests that about half of people eventually enter remission, but they can have late recurrences too. So one's much more cautious about weaning treatment and in counseling them about the prognosis. And then there are some devastating syndromes, which I'm not going to dwell on in childhood: Lennox-Gastaut, myoclonic astatic epilepsy, and Landau-Kleffner, which each of them carry a particular prognosis. And as I said, sometimes we don't know. So here's a CAT scan of someone I saw probably 20 years ago. This is just to give you an idea of the usefulness of imaging. So here's a large vascular malformation which caused very devastating seizures for this boy. We operated on him many years ago, and I still see him from time to time. He's required a little bit of medication, but he's remained seizure-free for better part of 2 decades, I think. This is an MRI scan and this is a particular MRI scan. It's now called a susceptibility-weighted image, but the idea is that this picks up tiny areas of either calcium or mineral. And in this instance, this person had had a severe traumatic brain injury and had epilepsy and had a multitude of these little abnormalities which were not apparent on CAT scan or on any of the other MRI sequences, but these were little old microhemorrhages that had occurred in relation to neuronal shearing in this diffuse traumatic brain injury. Now, this scan gives you an idea of how neatly we can distinguish cortex, which is the gray matter here, from the white matter. And our images, this is an old image actually from more than 10 years ago. Images today are even better than this. And we can see cortex, and we can see the white matter, and these are the temporal lobes. And this, you'll see, looks a bit like a seahorse, hence the term hippocampus. And importantly, in this person, this right hippocampus, the left is on the right and the right is on the left. The right hippocampus is much bigger than the left one, which is darker in appearance. And in this instance, this person had devastating, really severe epilepsy related to neuronal loss in the left hippocampus. Surgery produced a very satisfactory outcome here. Again, someone we operated on about 18 years ago. So the advance of MRI imaging in disclosing subtle causes of epilepsy is evident, and that's why I show you this. This is someone who previously had an abscess and had epilepsy. Here's another example of hippocampal atrophy. You can see the nice normal right hippocampus, and you can see the dark spore left hippocampus here. This is parahippocampal gyrus here. And this is someone who has had dual pathology, and we see this not infrequently. So here you'll see his left hippocampal atrophy. This one's right. And this person sustained an early insult in very early childhood, hypoxic injury with a stroke in this part of the brain. And interestingly, this woman had 2 types of seizures. We initially operated on this because this is where her seizures were starting. And she was seizure-free for many years. And presumably this had in some way led to the atrophy here. Many years later, she started developing a few seizures which originated here, which we treated successfully with medication. It's an interesting subject in itself of kindling, etc. This is someone who's got what looks like a horrible, nasty, big tumor spanning the entire temporal and occipital lobes. In fact, this is a developmental disorder of the brain. And this guy was in his 50s. He'd had epilepsy for most of his life. And when we studied him with intracranial investigations, we found that his seizures originated in the amygdala and the hippocampus, and a relatively restricted resection here produced a seizure-free outcome for many years. I haven't seen him for a number of years now, but he was seizure-free when I last saw him. Again, because of this, we left him on a little bit of medication, but this had produced absolutely no dysfunction cognitively, linguistically, or visually. I told you a bit about the malformations of cortical development. So here's an interesting one of someone who was undiagnosed for many years. The cause was undiagnosed. This woman had many MRI scans over many years. And you'll know that neuronal migration starts with neurons that are located around the ventricles and then migrate outwards. And you'll notice that just lateral to the right temporal horn here is a nodule of gray matter, and there's a slightly bigger one on the left here. And this is called periventricular nodular heterotopia. Sometimes you see a little bit of a band of some gray matter that's left in the white matter. And these circuits and systems produce seizures. And it's interesting that you can put depth electrodes into these, and sometimes you can see that the seizures originating here, sometimes here, and sometimes synchronously in both of them. And this is, again, someone who had many MRI scans. This is a sequence from the late 90s. And again, to emphasize, our images are much clearer now. But here was an area of cortical thickening. So this was what was termed polymicrogyria, a boy with very disabling seizures. He had a resection, and he also was seizure-free. How are we doing time-wise? We're right. So why do we bother with these classifications? Well, I think, as I said earlier, it impacts on prognosis, on counseling patients and their families, and then on treatment. So the prognosis of the epilepsies, and I again emphasize one shouldn't use the term epilepsy, or I don't like using it, is very variable depending on cause or syndrome. So if you take all these different types of epilepsies and you lump them together in one big basket, as it were. This was done by Anagers and Hauser in a very famous study published in 1979. Then overall, you find that about 2/3 of people become seizure-free, and seizure-free is defined as 5 years because you'll see the horizontal axis starts at 5 here. So about 2/3 of people with all these different types of epilepsies become seizure-free, and about half of them require medication to sustain that. Sorry, that should be 40%. But to emphasize, there are many different prognoses depending on cause or syndrome. So this is the basket of everything together. The other point about it, and the reason for showing this, is that people who enter remission do so early on. So if you're going to get better, if the epilepsy is going to go away, for the majority, the remission occurs earlier. And after that, the incremental probability of remission becomes smaller over time. More recent studies have shown that this becomes more complex. This pattern is more complex, and it parallels what we see. So there's some forms of, say, the genetic forms of epilepsy where people might be seizure-free for many years, and then in their 50s go through a patch again of seizures, etc. So this is a little bit of, to emphasize, this is not a little bit, a huge oversimplification of a very complex subject. The pattern in childhood, the childhood epilepsies looks similar, but for the fact that the graph and the trajectory of the graph is a lot flatter. And the reason it's a lot flatter is that in childhood, you have a disproportionate representation of these genetic forms of seizures. That's when they're most likely to manifest. And when they present, ones like absence seizures or the genetic generalized seizures or benign focal epilepsy, remission might take a number of years. And hence, the curve is not as steep as I showed you. But overall, you end up with about 70% of people who are seizure-free by the time they enter adulthood. Again, to emphasize, cause is important, syndrome is important. So you can see, for instance, kids who've got something pre-existing wrong in the brain, their prognosis is not terribly good, and the vast majority of them have a poor prognosis. So really, just to summarize, for most people, remission prognosis is good, but medications are often needed to required to sustain the remission. And in childhood, you often have to be a bit more patient to achieve that remission. Now, perhaps the most important message that one might get across today is there's nothing cool about having one seizure, let alone lots of seizures. It really is very, very harmful to patients' quality of life. And there's a huge body of literature to the impairment of the quality of life of people who, after all, some of whom only have a couple of seizures that last a minute or two. And it's always fascinating to sit and listen, sad in a way, to how sometimes people's lives are destroyed by a few seizures. A lot of it relates to the way we as society perceive, and family members perceive, these individuals, and the rules and the way we treat them, and the rules that we contrive to limit any damage, as it were. So if you talk to people about their quality of life, one of the most often, amongst adults, the one that's raised most often is the prohibition on driving. Uber and taxi services have somewhat improved that, but one's whole identity and sense of independence is contingent on driving. Stigmatization remains an enormous problem throughout the world. Patients who have seizures often live in constant fear of seizures. So just the odd seizure is enough to leave people with enduring fear. And that sometimes prompts us to even treat people who have the odd seizure with surgery in order to get their quality of life right. People fear losing control. They're embarrassed about having seizures. Some patients sustain very nasty injuries during seizures, horrible burns requiring plastic surgery, facial burns, lacerations, cosmetic injuries, fracture dislocations, fractures of the vertebrae, etc., etc. While there are labor laws, patients are discriminated against in the workplace. This is a relatively recent study about stigma in Europe, and you can see that stigma is very common. And other parts of other components of patients' quality of life are also impacted. Patients lose, if they're self-employed, they're less productive. Seizures impact on relationships. Patients feel horrible after seizures. And perhaps the most important is that mood symptomatology often accompanies the seizure disorder when the seizures are impacting on quality of life. And that in itself, mood has been shown to be has been shown to be the variable most strongly predictive of someone's perceived quality of life, someone who has a form of epilepsy. So what treatments have we got? Well, in principle, there are 4 broad categories: medications, which are necessary for most patients, operations when medications don't work. Occasionally, we do use something called the ketogenic or the modified Atkins diet, and occasionally we use the vagal nerve stimulator. We can talk about those, but there really isn't much time. I'm not going to spend any time, because I think we're out of time, on the different medications and the focal versus the generalized seizures, but just to say that cost is important, side effects are important, and number of times you've got to take it a day are important in terms of compliance. I spend an inordinate amount of time working through sequential changes in medications for patients in order to optimize quality of life. And quality of life involves getting someone seizure-free, but not at the cost of side effects. And the idea that medications inevitably produce side effects is flawed. We have a multitude of medications, and you can generally find a dose and a medication that doesn't produce side effects and affords people a good, normal quality of life. So that has to be the goal of treatment. I'm just going to skip on a bit here. So what happens when someone's seizures carry on medication? If seizures continue on treatment, then we have to reconsider the possibility, and one has to do this all the time, that your diagnosis is incorrect. Remember the first slide? Seizures may be mimicked by other things, and someone who doesn't get better on treatment, you have to ask, is your diagnosis correct? While we're about it, if there's diagnostic uncertainty, it generally is not a good idea to put someone on treatment for epilepsy until you're certain. The reasons, again, are that you might incorrectly treat someone for decades because diagnosis and treatment sticks. And secondly, response to, so the treatment, if someone's seizures stop, doesn't prove that it was seizures. The seizures might have stopped by themselves. There might be a placebo response, and the other neurological conditions that respond to anti-epileptic drugs, such as migraine, etc. Second reason that someone might continue to have seizures is that they might stop taking their treatment. And the reason they stop taking their treatment is generally for a very good reason. They get side effects. The doctor hasn't discussed the side effects. They can't afford the medication, or they don't have access to the clinic, etc., etc. And if someone has failed treatment, and we can talk about that in itself, then the option, once we've exhausted medications, is to consider surgery, especially people who have so-called focal forms of epilepsy. So there was a randomized trial from the center where I was at. In fact, the study was done while I was there, a randomized controlled trial, which showed that temporal lobectomy is markedly more effective than continuing with medical treatment for people who have seizures that continue despite trials of 2 drugs. And you'll see a year after randomization, only 1 patient in the medical group was seizure-free, whereas 2/3 of patients in the surgical group were seizure-free. And this was a very complex cohort of patients. Just to re-emphasize neuroimaging and how useful it can be in identifying cause. Here's hippocampal atrophy again. This is someone I saw in 1999. I still know him now socially, and he was a teenage boy who had devastating seizures, which started in his medial occipital lobe. He had a malformation of cortical development here. He'd had seizures for a number of years, and following a surgery in this region, which produced absolutely no dysfunction on formal visual fields, there was a very minimal peripheral visual field loss, but that was it. And he, 20 odd years later, is seizure-free, married, has children, and functioning independently, and runs his own IT business. Now, how do we work out what's going on in the brain? Well, it's a complex process. So you start with history, and history, and history. You have to get a multitude of histories about what goes on in seizures, and that's a discussion itself. We then do video EEG recordings, and that is a whole discussion itself, which I don't have time to explain. And then sometimes we have to go in and actually put electrodes on the surface of the brain and work out where seizures are. All of this is to localize regions of seizure onset. So this is someone who, in fact, was seen somewhere else in another city, who had video EEG recordings. His EEGs were completely normal during many seizures, and he was sent to a psychiatrist and diagnosed as having non-epileptic seizures. And the clue in him was in the history. He had epilepsy, and he came here, and we put in these electrodes. So this is the top of the head, this is the temporal lobe down here, this is the Sylvian fissure, so he's upside down, as it were. We did video EEG recordings on him. His region of seizure onset was here. We were through those electrodes that are in place here. We were able to do cortical stimulation for language functioning and for motor functioning, and sometimes for other functions too, while someone's in the ward on monitoring. And we were able to show that his posterior language area was in this region here. And so we were able to do an operation of his left superior temporal gyrus and part of his left mid-temporal gyrus, quite a long way forward. So you can see this is a large resection. sparing language cortex, and he remains seizure-free today, many years after having had his operation. There's the particular myth about seizures that the brain is being deprived of oxygen during a seizure, and one can sometimes use SPECT scanning to localize seizure onset. It's not a great test because the time that it takes, you have to have someone sit there, you have to inject the radionucleotide, which is made up beforehand. The time from which it injects to the time in which it gets to the brain is of the order can be up to 90 seconds, by which time seizures sometimes propagate within milliseconds, let alone seconds, to different parts of the brain. But sometimes it's useful in localizing seizures, and this is one we did many years ago, which actually co-localized to a developmental abnormality in his left posterior temporal region. And the point of showing this to you is that, in fact, there is hyperperfusion in this area where the seizure was. We obviously have to make sure that the operation is done safely. Remarkably, for many patients, you can do an operation and, in fact, improve cognitive functioning. And it attests to the detrimental effects of frequent seizures and frequent discharges on cognitive functioning. So to give you just a typical example, I remember years ago a little boy of 10 years old who had left temporal lobe epilepsy. He was top of his class. His cognitive functioning declined dramatically once his seizures started. He had a developmental disorder in his left mid-temporal region. We did an operation on him. Immediately post-operatively, he was mildly, his dysphasia was a bit worse. And then he recovered, and his trajectory was again, he ended up functioning within months way above what his mother had predicted. We have many other examples or anecdotes where people immediately after the operation are functioning cognitively at a better level than they did beforehand. So the idea is you have to work out beforehand what's going on cognitively with someone and what's going on behaviorally, and that's part of the fun of epilepsy surgery. You have to understand that. You have to understand what happens in that part of the brain that you want to operate on. And then having done that, you can then plan your operation. And in thinking and understanding that, we rely on neuropsychological testing. In the old days, we used to do something called a WADA, or a sodium amytal test, where half the brain was paralyzed for a few minutes. You tested language and memory function on the other side. The person still stays awake while you're paralyzed half the brain, by the way. And then you do the converse. As I said to you, we stimulate the cortex both while patients in the ward, and we wake them up during operations. And we can do something called functional MRI, where you put someone in the MRI scanner, and when they use a particular part of their cortex or their brain, the perfusion to that part of the brain increases. So if you do finger tapping, as this paradigm required, when you do finger tapping, the part of the brain that mediates that finger tapping is activated, and in so doing, it requires a little bit more blood. Perfusion goes up by 2 or 3%. Blood is paramagnetic. You can measure it and do a subtraction from comparing the movement to a period, a basal state. And then in this instance, here we had a person with a developmental disorder immediately behind a motor function. This is the right, so it's the left hand. And we were safely able to subsequently put in electrodes, know that motor function was in front of the lesion. How are we doing? I think we're probably out of time, I would imagine. So I think maybe I won't talk about status epilepticus at this point. I think I'll end off there, and we can always talk about it if there are questions about it. But I think I've said enough. Are there any questions or comments? Or?
I think there was the question from earlier in the comments. Where do you think doctors in South Africa fail in terms of treating and diagnosing epilepsy?
Yeah, I think that's a fantastic question. So it's not just in South Africa, it's right across the world. It's called the treatment gap. And believe it or not, there's a treatment gap in every country in the world. There are attempts, and there've been many attempts at systematically studying the treatment gap. And And so even in developed countries, there's a significant treatment gap. So, so, you know, if you ask where do you apply your resources into improving treatment outcomes for people in South Africa with epilepsy, well, it's diagnosis, so you need more healthcare workers. It's treatment, so you need more widely available medications. You need more specialists, so you would need more neurologists. We have a limited number of neurologists. You need a greater availability of some of the newer anti-epileptic drugs, which are more expensive, and ideally you need even much more wider availability of neurosurgery. Even in very developed countries like America, there are huge treatment gaps when it comes to things like epilepsy surgery, let alone anti-epileptic drug therapy. So I think the answer to your question is multifaceted. I think, you know, some countries in Africa have successfully embarked on training nurses and healthcare workers in the recognition of seizures. So, and modern, I think it's an area which I think we're going to see a lot of development in, is the area of telemedicine, and potentially with cell phones. We can now, I use this often for perhaps the majority of my patients, but they can video seizures using their cell phones and send those electronically. And that is a huge help in terms of diagnosis, and also in distinguishing sometimes between epileptic and non-epileptic seizures. So technology offers us all sorts of opportunities in Africa, I would have thought. I think there are a lot of other comments related to that, but I don't want to digress into all the little details. I think enough said.
And then I just have a question in terms of, you were talking about epilepsy surgery, the temporal lobectomies. Is it sort of a standard surgery, or is it more, so you only take out the area involved in the seizure?
Yeah, so our approach has been very much one of tailored temporal lobectomies. More often than not, seizures originate in the medial temporal lobes, but seizures can be neocortical, and sometimes they can involve large parts of the temporal lobe. Sometimes they can be relatively restricted to the medial temporal lobe. So we do those often, and our approach has been very much one of a tailored approach. In principle, the world has had kind of two big operations. One is called a standard temporal lobectomy, where about 4 or 5 centimeters of the neocortex is taken in addition to the medial temporal lobe. And other people have taken a more selective approach, which is surgically a lot more tricky, where for people with just medial temporal lobe epilepsy, you're largely removing amygdala, hippocampus, parahippocampal gyrus, and entorhinal cortex, etc., piriform cortex. We've just completed a randomized controlled trial in it, which we've submitted for publication, and hopefully you'll see that in due course. It's an interesting discussion in itself. But we do a lot of in our center here, we do an awful lot of extra temporal operations as well. So in different parts of the brain, frontal, parietal, occipital, and then the patients who have the devastating drop attacks, generalized seizures, and corpus callosotomy, where you section either the anterior 2/3 or the entire corpus callosum, is something that we do several of per year. Sometimes we do big regional operations. We probably end up doing 1 or 2 hemispherectomies per year, where patients have severe devastating diseases which render the hemisphere severely dysfunctional, produce multifocal seizures. And remarkably, in these patients, and I have one patient who I think still the only patient in the world who had a normal MRI scan who we did hemispherectomy on. And what was quite striking in him, just to give you an example, was the night after the operation. This was a boy who was sitting there drooling, having seizures all the time. Straight after the operation, he was sitting there bright-eyed, chatting away, drooling had stopped, and he was brighter than either his family had ever seen him, which just tells you something about the effect of the ongoing seizures on the rest of his brain. And we see that often with more localized resections as well. Adjacent cortex becomes dysfunctional secondary to this, seizure process that goes on in cortex.
And then following on from that, I assume before you do the surgery, you obviously discuss the side effects and the deficit that the patient will have after you remove whichever part of the brain. I assume you then balance the sort of advantages versus disadvantages of having the surgery. What kind of, obviously it depends on what part of the brain you're removing, but is it often that the patient has large deficits post-surgery, even though they might not, the epilepsy has been cured?
Yeah, the short answer is no. You're doing surgery not to stop seizures. You're doing surgery to improve someone's quality of life. So we bring patients in here. Typically I spend 1 to 4 weeks thinking of their problem through, talking to them and their family about their quality of life, and then thinking very carefully about what surgery will do for them, not just immediately post-operatively, not just immediately in terms of seizures, but in terms of the trajectory for the rest of their life. And there has to be a huge probability that you're going to, or a substantial probability that you're overall going to produce a marked improvement in someone's quality of life. Otherwise, why invest in a process which has small inherent risks? And by and large, when we're doing operations, well, we never do operations where there's significant risk. I think that's the bottom line. Occasionally, there's small little issues that you have to discuss. For instance, if you're doing a dominant temporal neocortical resection near language area, patients sometimes in the early post-operative period have a slight increase in word finding difficulties. Sometimes that endures. But if we offer someone surgery for that, the little loss that they might incur has to be offset by a huge probability of being seizure-free and a huge probability of a gain in their overall quality of life. So I emphasize quality of life, and quality of life involves seizures, but functioning, it involves mood, it involves going through all of this, all of those things. So it's really a question of thinking holistically, and cognition and behavior coming to that very important.
Thank you. I don't see any other questions. So in closing, I'd like to thank Dr. Butler and Dr. Raimondo for taking the time to share their knowledge with us. It's been a very interesting and educational session. I'd also like to thank all our members, Cortex Club and MedSup members alike, for joining us this afternoon. It was great to have so many people here and interacting with the material. Erin, if you have nothing else, I think we'll close, and I hope that everyone has a wonderful day further. Thank you, Dr. Butler, Dr. Raimondo again.
Pleasure. Thank you very much.
Thanks, Dr. Butler. Cheers, everyone. Thanks for coming.